Hi, Assalamualaikum. My name is Siti Hajar binti Hasbullah. I am year 1 from KPT. My metric number is 22303812. Today, I will present about anatomy of ear. Our ears are divided into three parts. First of all, the outermost part is the external ear, then middle ear and the inner ear. First of all, the yellow color is a temporal bone. As we can see here, the anatomy for the external part start with the helix and lobule. Both of these is a auricle that have a fan or a cape shape. It is a cartilaginous. The structure that I highlight with the blue color is a external auditory canal. External auditory canal is a short, curved tube about 2.5 cm long, lined with the skin-bearing hairs and sebaceous glands, and also cereminous glands. It lies in the temporal bone and leads to the tympanic membrane. Our tympanic membrane, also we known as is a eardrum. Eardrum is a thin and semi-transparent partition between the external auditory canal and middle ear, covered by epidermis and lined by simple cuboidal epithelium. Anatomy of the middle ear, as we can see, these tiny three bones that I highlight with the red color is an auditory ossicle. The first bone that attached to our eardrum is a malleus or hammer. The bones that connect malleus with other bones is incus or anvil, and the last bones that attach to oval window is steps or stirrup. This tree is auditory ossicle. The path leading down that I highlight with the red color is known as the extension tube. Extension tube is a tube that extends from the middle ear cavity to the pharynx or a throat. Then we will talk about muscle at our middle ear. This is a tensor tympanic muscle. It originates on the petrous portion of the temporal bone and insert on the malleus. I will show you petrous portion and temporal bone. This is our temporal bone. The yellow color that I highlight is a petrous part. And then another muscle is a stapedius muscle. Stapedius muscle, this muscle is insert on the stapes. I will show you that this is a stapedius muscle and it attached with a stapes. We will go more detail about our inner ear. There are three structures that are part of our inner ear. The first is a semicircular canal, cochlea, and the vestibule. This is a bony labyrinth. Bony labyrinth filled with a perineum fluid. This is a membranous labyrinth. Membranous labyrinth filled with endolymph fluid. Both of this fluid, the function of this fluid is to conduct sound vibration, convert sound wave to nerve impulse, and detect the linear and static equilibrium. Also, the function is to detect the dynamic equilibrium. This is a ampulla of semicircular duct. The red color is a oval window, and this is a secondary tympanic membrane in a round window. So we will focus more on our cochlea. As we can see, this is the structure of our cochlea. Cochlea is a long tube curved into a shape that resembles a snail's shell. Function of cochlea is to convert sound wave into nerve impulse. This is a tiny three bones in the middle ear. That is malleus, incus, stapes at oval window, and then we have round window, external auditory canal, tympanic membrane or eardrum, middle ear cavity. The blue color is the helicotrema. Cochlea duct is to contain endolymph and spiral organ of Corta. Scala vestibule. Scala vestibule is filled with perilymph. the floor of the scala vestibule is formed by the vestibular membrane. And then scala tympani. Scala tympani also filled with perilymph, but the roof is formed by the basilar membrane. The main structure of cochlea that I highlight is scala tympani, scala vestibule, and cochlear duct. The last part of the ear is a spiral organ of Corti. A spiral organ of Corti is a structure made of several membranes and collections of cells which extend the length of the cochlear duct. The blue color is a tectorial membrane. Tectorial membrane is a project into cochlear duct and rests on top of the hair of a hair cells. And the yellow color is outer hair cells. This is project hairs upward into the inferior surface of the tectorial membrane. The pink color is the inner hair cell, and the last part is the purple color is basilar membrane.
Hi, Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Isam Rosli. I'm from degree nursing year 1, and today I'm going to explain about physiology in hearing and smelling. The first one is transmission of sound wave. Sound waves are collective by the pinna and focus into the auditory canal. The vibration pass down the canal and strike the tympanic membrane. Okay, what we know from here is the sound wave is collected by the pinna. This is the pinna, this is our outer ear, and then focus into the auditory canal. Auditory is our tube as our hearing in our ear, as you can see at the slide, and then something. Okay, membrane vibrate in response to the sound wave and move the malleus. The malleus move incus, which in turn move the step. The step rests on the membrane across the oval window. This membrane vibrates in response to the vibration carried by the step. Okay, which means that malleus must. So you can see here, the malleus move the incus, which means that the malleus get the sound energy from outside into our malleus, and then it's move the incus and vibrate the step, and then after that it send the sound to oval window. Oval window transmits sound into the scala vertebrae. Second one is sound to impulse. How is sound wave convert to impulse? Okay, the receptor of the organ of Corti, the hair cell, as known as mechanoreceptor, have hair like called stereocilia. The vibration move the basilar membrane and hair cell against the tectorial membrane. This movement leads to the displacement of the stereocilia. Okay, the fibrous connection mechanically open cation influx of ion is called depolarization, and this trigger open voltage gauge, calcium channel, and near the base of the cell is known as neurotransmitter release. And we know the function is neurotransmitter is release. Bending the hair cell is in the opposite direction. Close the channel is called as depolarization. As the sound wave vibrate the basilar membrane, stereocilia are bent back and forward. One is auditory pathway. Cochlear nerve form the cochlear branch of the vestibular cochlear nerve. This axon intermedulla oblongata and synapse at the cochlear nucleus. Okay, the information cross to the opposite side of the brain and ascend to the inferior colliculus of the mesencephalon before reach the cerebral or cortex ascending auditory synapse in the medial geniculus nucleus of the thalamus. Okay, projection fiber then delivered the info to the auditory cortex of the temporal lobe. This is the last part, which is equilibrium. Best way of equilibrium sensation. We have four functions of the vestibular nucleus. The first is sensory information for the equilibrium and balance. Relay information from vestibular apparatus to the cerebral cortex head, position and movement, and sending command to the motor nuclear in the brain system and spinal cord. So that's all that I have to explain about this, and I hope you all can study well for this topic.
Hi, Assalamualaikum. My name is Afifa Binti Abdul Samad, and I'm from Science Forensic Year 1. So let's move on to the anatomical part of the sensory system of taste and smell. For the taste or gustation system, the main organ is our tongue. Do you know that the receptor organ of the taste system is the taste bud? The receptor of taste sensation are located in the taste bud found in elevation on the tongue called papillae. Papillae provide a rough texture to the upper surface of the tongue. There are three types of papillae that contain taste bud. The first one is circumvallate papillae, the next is fungiform papillae, and the foliate papillae. The taste buds are located on the tops of fungiform papillae and on the side walls of foliate papillae and also circumvallate papillae, while filiform papillae here do not contain any taste bud. Each taste bud has three important epithelial cells in its plus-shaped body. The first one is the supporting cells, which surround about 50 gustatory receptor cells. The second one is the gustatory receptor cells, which project along gustatory head to the external surface through the taste pore, the opening in the taste buds. These cells are actually separate receptor cells. They do not have an axon, but rather synapse with dendrites of the first-order sensory neurons of the gustatory pathway. The third one is basal cells. Basal cells are stem cells that produce supporting cells which develop into gustatory receptor cells that have lifespan about 10 days. Done with the taste system, we are moving on to the smell or olfaction system. The main organ is our nose. The first important olfactory organ is the olfactory epithelium, which occupies the superior part of the nasal cavity, covering the inferior surface of the cribriform blade, and it is extending along the superior nasal concha. It contains olfactory receptors, supporting cells, and basal cells. The olfactory receptors is the first order of the olfactory pathway. It projects several cilia called olfactory hairs from a knob-shaped tip on each olfactory receptor. Olfactory hairs, which is the chemoreceptors, is parts of olfactory receptors responded to inhaled chemicals. Chemicals that have odor can stimulate the olfactory hairs. The axons of olfactory receptor extend from the olfactory epithelium to the olfactory bulb. Next, the supporting cells is the columnar epithelial cells of the mucous membrane lining the nose. It provides physical support, nourishment, and electrical insulation for the olfactory receptor. It also helps detoxify chemicals that come in contact with olfactory epithelium. After that, the basal cells are stem cells located between the bases of the supporting cells. These cells continually undergoing cell division to produce new olfactory receptor which live for only a month or so before being replaced. Enough of the olfactory epithelium, let's go to the second olfactory organ, which is the lamina propria. It is an underlying layer of loose connective tissue containing olfactory glands are called as Bowman's glands. These glands will produce mucus that moisten the surface of the olfactory epithelium and serve as solvent for inhaled odorants. Lastly, is the olfactory bulb. This organ is highly organized structure composed of several distinct layers and synaptic specialization. In the olfactory bulb, there are layers from outside towards the center of the bulb. It is differentiated as follows. It starts with the glomerular layer, followed by external plexiform layer, mitral cell layer, which contain mitral cells, internal plexiform layer, and it ends with granular cell layer. For your information, mitral cells is the second order neurons contacted by the nerve fibers at the glomerular layer of the bulb. Each mitral cell is contacted by at least 1,000 olfactory nerve fibers.
Hi guys, I'm Afika and welcome to my presentation on the fascinating world of olfaction and gustation physiology. In this presentation, I will talk about the transduction process and the pathway that involve in the olfaction and gustation. So for the first part that I want to talk about is olfaction, which is our sense of smell. So within our nasal cavity, we have thousands of tiny hair-like structure called olfactory receptor. When we breathe in, odor molecules enter our nasal cavity and bind to this receptor, triggering a chemical reaction. This chemical reaction leads to the transduction process, where the odor molecules is converted into an electrical signal. This transduction process is not only occur in the olfactory, but also occur in the gustatory part that I will explain later. But this transduction process, I will not repeat again in the gustatory because it is a similar process. So, what is this transduction process? This transduction process is occur when the stimulant, which is the odor or the testin, is bind with the binding protein in the membrane of the receptor. So when the stimulant is bind with the binding protein, the adenylate cyclase, the enzyme that will convert the ATP to CAMP will be activated because of the stimulant bind with the binding protein. And then the CAMP will open the closed sodium channel. It will open the cation channel, and when the cation channel open, the cation, which is the sodium potassium, will pass through the cation channel. So it will cause the depolarization and initiate the electrical signal. So these electrical signals are then sent to our olfactory bulb located at the base of the brain. From the olfactory bulb, the electrical signal travel along the olfactory nerve pathways to different regions of our brain, including to the primary olfactory area in the temporal lobe of the cerebral cortex, where conscious of awareness of smell begins, and to the hypothalamus and the limbic system, which is associated with emotion and memory. That's why certain smell can instantly transport us back to a specific memory or evoke strong emotion. The connection between our sense of smell and our brain is truly remarkable. So now let's turn our attention to gustation or our sense of taste. The taste bud, where present on our tongue, play a crucial role in this sensory experience. Each taste bud contains taste receptor cell, which can detect the 5 primary tastes, which is the sweet, sour, salty, bitter, and umami. These receptor cells are responsible for the transduction process, converting taste molecule into electrical signal. It's similar with the olfactory part before. So for the gustatory part, the electrical signal generated by the taste receptor cell are transmitted to the brain through cranial nerve, specifically the facial, glossopharyngeal, and vagus nerve. So this electrical signal reach different regions of the brain, including the gustatory cortex, where they are interpreted as different taste and flavor. So it's important to note that our sense of taste work hand in hand with our sense of smell. In fact, around 80% of what we perceive as taste actually comes from our sense of smell. So the next time you savor a delicious dish or enjoy a pleasant aroma, remember that it is the combined work of our olfaction and gustation senses, as they work together to create the incredible flavors we experience. And our sense of smell and taste are not just about enjoying food, but also serve as crucial survival mechanism. They help us detect spoiled food, recognize potential dangers, and avoid harmful substances. And I think that concludes my explanation on olfaction and gustation physiology. I really hope everyone who are watching this gain a deeper understanding of these senses and their intricate process of transduction and pathway. So that's all from me. Thank you.
Now I'll be explaining to you the human vision, or known as the anatomy of vision. Let's start. In this topic, it consists of 5 contents. The first one is introduction of eyes, second is external anatomy of the eyes, third structure of the eye, fourth photoreceptor cells, and fifth the eye chambers. Starting with the introduction, what is eyes? Eyes is a pair of special sense organs that works with the brain to provide us vision, situated one of each side of the nose in the upper face. So this is the upper face, and each one on each side, left and right. They occupy 2 bony openings, here and here, on each side of the skull. Want to know more about the eyes? Okay, eyes is embedded in orbital fat and covered by fascia sheath of the eyeball. Spherical in shape, and the diameter is 24 millimeters. Consists of 3 layers, which is the outer layer, the middle layer, and the inner layer. The outer layer has sclera, cornea, pupil, and lens, while the middle layer has iris, ciliary body, choroid, and lastly in the inner layer, has retina, optic nerve, and vitreous humor. Vitreous humor is the chamber for the posterior part of the eye. Alright, so let's take a look of the external anatomy of the eye. External means what we have on the face, on the area of the eye. Starting from here, we have the eyebrow, and then the upper lid. We have the eyelashes, sclera, this white part, and also known as the outer layer of the eyes. Then we have the iris, which is responsible giving our eyes the color. Pupil, this dark spot, the black spot here. And then there is lacrimal caruncle, this little hole here where our tears come out. And then the very edge here, known as medial commissure, the very edge near the nose. And then there is also near the ears, known as lateral commissure. And then there is lower eyelid here. Let's move on to the structure of the eye, starting from sclera. Sclera is the white part, the outermost layer, which is here, 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 basically the white part. Okay, I'm sorry, here. It is tough. It is tough. The function is to protect and give shape to the eye, serve as insertion point for external muscle, and move eyeballs within their socket. Sclera anterior surface is specialized into cornea. Now, on the second layer, we have choroid. Second layer is here, the brown reddish layer. It is dark brown pigmented to absorb light. It is specialized into the iris, associated with eye color, ciliary muscle, and the zonular fiber. Where is the zonular fiber? Zonular fiber is this strand that holds the eye's lens, and actually there is one right here. It is also known as zonular fiber. Finally, the innermost layer, which is known as retina. Retina lining the inner posterior surface of the eye. What is posterior? Posterior meaning is on the back of the eye, containing numerous types of neurons and photoreceptor cells. Features of retina can be viewed through the pupil with an ophthalmoscope. From the ophthalmoscope, there is three things that can be seen lying in the surface, on the inner surface of the retina, which is fovea centralis, optic disc, and blood vessel. Where is fovea centralis, where is optic disc, where is blood vessel? So this is the fovea centralis, this spot is the optic disc, and this is the blood vessel, these red strands. Photoreceptors. Photoreceptor is a specialized cells of the eye that detect and convert light into action potentials. There is two types of photoreceptors called rods and cones. Why rods and cones? Because look at the shape. Resemble rods, and this one similar like a cone. Photoreceptors cells can be divided into three segments, which is the outer segment, the inner segment, and the synaptic terminal. Outer segment contains special photosensitive chemicals that trap light and convert it into action potential. Inner segment, however, has nucleus and responsible for synthesis of photopigments and energy production. Synaptic terminal contains bipolar cell that carry action potential to other retinal cells. Alright, so let's differentiate between rod-shaped photoreceptors and cone-shaped photoreceptor. For the rod-shaped, per retina there is 120 to 140 million rods, and it is sensitive to light rather than color. Responsible for black and white vision, meaning it is work best in dim light or no light at all. A single photon can excite a rod. In bright light, rod cells are non-functioning. How about the cones? The cones, 5 to 6 million cones per retina. It requires bright light to function, at least 100 photons, responsible for color vision. Most of the cones are found in macula luteal, which is located on the central area of the retina. Highest density of cone is located in the fovea centralis, which has the highest visual acuity, meaning on the area it can create sharpest visual. Alright, so the eye has two chambers. The first chamber is known as aqueous humor. Aqueous humor is located on the anterior cavity. Look at this diagram. This is the anterior cavity between the iris and the cornea. So here's the chamber, and the chamber is filled with water-like fluid similar to blood plasma. Now, the second chamber is known as vitreous humor. It is located on posterior cavity. Here is the posterior cavity, which is between the lens and the retina, and it is filled with viscous jelly-like substance.
Let's continue with physiology of our vision. It will consist of three parts, which is focusing light on the retina, accommodation reflex, and photochemical activity. Let's take a look on the first part, which is focusing of light on the retina. So, when we look at an object, the light reflected from the object enter the eye through the cornea. The cornea bends light before it passes through a watery substance, aqueous humor. The light continues to pass through the black hole in the center of the iris, the pupil. The pupil then automatically gets bigger and smaller depending on the intensity of the light. The light penetrates the pupil and reaches the lens behind it. The lens adjusts its shape to bend and focus the light a second time, giving you a clear image of what you are looking at. At this point, the light was refracted twice, from the cornea through the lens and then from the lens to the retina. This double bending has actually flipped the image upside down. The light then travels from the lens to the vitreous back of the eye until it reaches the retina. The light is then focused on the retina, which contain nerve cells called photoreceptor. While light is focused throughout the retina, most of light entering the eye is concentrated at the central point in the retina. The retinal nerve collects all the electrical impulses which are transmitted via the optical nerve to the brain. Next, we will move on with second part, which is accommodation reflex. What is accommodation reflex? The accommodation reflex is the process by which the eye adjusts focus to obtain a clear image of object at different distance. This involves changing the curvature of the lens to focus on distant to close or distance object. Accommodation is a reflex action that involves changing the lens to focus light rays from an object perfectly on the retina. The image created behind the retina when viewing an object that is less than 6 meter distance, but because of the lens accommodated, an image formed in the retina allowing us to perceive the object. The ciliary muscle tighten and lens thicken to allow accommodation to focus on a closer object. Similar to this, when viewing a distant object, the ciliary muscle will relax, increasing the ligament tension and pulling the lens, causing the lens to narrow and generate an image in a retina. This is the last part of the physiology of vision, which is photochemical activity. Are you wondering how is EPI created from light or photons? The substance known as photopigments are present in the photoreceptor to absorb light. There are two parts of this, which is processes favored in the dark and the processes activated by the light. Rhodopsin are embedded in the disc structure membrane in the outer segment of the photoreceptor cells. When the membrane-bound enzyme guanylyl cyclase is not active, GTP is converted to cyclic GMP, which is cGMP, the second messenger molecule. The ligand-gated cation channel in the outer segment membrane is kept open by the cGMP because of the continuous inflow of the calcium and sodium. High photoreceptor cell that is kept in a depolarized condition in the dark. Rhodopsin is composed of two components that are joined together, which is opsin and cis-retinol. Photon in the current lights impact the 1,1-cis region of the rhodopsin and transform it into all trans. Not all trans can remain attached to opsin. Opsin interacts with the G protein family members, which is transducin. Phosphodiesterase is activated by transducin, and this enzyme quickly breaks down the cGMP, reduced level of the cGMP, which cause the cation channel to shut and the membrane potential hyperpolarize, block all the release of neurotransmitter